Hello, I am Dr. Williams, and I will be providing your lecture on adaptive immunity for your advanced pathophysiology class. All the information in this lecture can be found in Chapter 8 in your textbook. It would be beneficial for you to utilize your textbook along with the slides to help enhance your knowledge related to the content that will be introduced to you. The content in this lecture is just the beginning of the information that is important for you to know. So please use this lecture along with your textbook to gain a better understanding about adaptive immunity. I also want you to note that we have been talking about the immune system in sections. However, note that the immune system, if it is an innate immune system or the adaptive, that they work together. They're synergistic. Although we are talking about them in sections, when we talk about what is actually going on in the body, they work together. So we're going to get started talking about adaptive immunity, but I would like to just kind of do a recap on some of the important parts related to the inflammation or inflammatory immune response that we have, so that as you're listening to this lecture and reading your textbook about adaptive immunity, you can see how things differ. So as we've discussed in the previous lecture, when you think about inflammation, think about a process where the components needed for inflammation, they are already in our body. They already exist in the blood and the tissues. And then they are activated immediately once the body is exposed to like some microorganism. Also, when you think about the inflammation, think about the response that occurs with inflammation. It will occur in the same manner despite what the organism that started the inflammation process is. It is not specific. It is a general response to any microorganism or pathogen. And then the third thing I want you to think about is once the inflammation process occurs, the residual that is left as part of the inflammation process or that is produced because of the inflammation process, it must be removed immediately so that we could have continued healing of the tissue and that it will not cause any more damage to the surrounding areas. And the final thing I want you to think about when we talked about inflammation is that no matter what the microorganism, you're going to get the same response. So now let's talk about adaptive immunity. I know that I said we would start to talk about adaptive immunity, but before we get started with the actual lecture, I have chosen to include in this lecture deck kind of an outline as you listen to all the information, and it has so much content, kind of an overview outline of what you should be trying to grasp as you hear a lot of information with a lot of details. So if you follow this outline, it will help you know the basics of what will be expected of you from this class. And for those of you who are so excited about how adaptive immunity functions, please refer to your textbook where you will get a lot more detail. Also, when you listen to the slides, I have given you the general overview of each one of these 5 sections. However, you can go to your textbook yourself and get the details and fill it in. I hope this is helpful as we begin this journey on adaptive immunity because it has a lot of content, but I hope this helps you to categorize it and understand what you should try to retain. On this slide, you can see what the objectives that we will cover during this lecture. I started the lecture off doing a recap of the inflammatory response that occurs in our body. However, now I want to talk a little bit about the adaptive characteristics, some of the general characteristics that we'll see. When we talk about the adaptive immune system, we're talking about a part of our immune system and our immunity that develops slowly over time. We're also talking about a immune response that is inducible once it has been exposed to things like lymphocytes and antibodies. And then once it has been exposed to lymphocytes or antibodies, then what it will do is it will produce a response to a specific infection. The other general thing I want you to know about the adaptive immune system is that the adaptive immune system only responds to specific antigens or agents. Okay? And then finally is the adaptive immune system has a memory. So you should be able to see the difference between the inflammatory response and what happens when we have adaptive immunity. So if you remember, we talked about adaptive immunity having a memory. So what we know is with that memory, we get these end byproducts, which is the T and B cells, which are lymphocytes. And then we get these antibodies that are going to be part of that memory that can then be utilized anytime your body is exposed to any type of pathogen or microorganism. So that's kind of the overall picture of the adaptive system. Now let's talk specifically about how that process actually works. Let's talk about those steps in the adaptive process. The adaptive process or adaptive immunity has two stages: clonal diversity and clonal selection. Clonal diversity begins when we are born. Two phases of adaptive immunity. First phase.
So let's now talk a little bit more about the two phases of adaptive immunity.
It occurs.
Let's start with the first phase, which is the clonal phase.
Organs, the thymus,
or clonal
and the
diversity.
Bone marrow. Hence the names T cells and B cells.
I would like to
They
utilize
are organ-specific
this image
cell
to
surface
talk
proteins
you through
that identify them, and they're on the surface of the antigen
adaptive
receptors.
immunity.
And they will then recognize one specific antigen.
I would like to use
even
this
before
image
they
to walk you
encounter
through the
that
phases
antigen.
of adaptive immunity.
So they're able to recognize
Let's start with
them,
the
although
clonal
they've
diversity
never
phase.
encountered them. They call
Clonal
them
diversity
naive.
begins when we are
And then
born,
what will
or
happen
before
is
we are born, when we are
they
fetuses.
will then move from the
And what
primary
occurs is in the thymus and the
lymphoid
bone marrow,
or central
which are the
organs,
primary
which was the thymus and the bone marrow,
organs. That's where
to
this
the liver
starts.
and the
Those
lymph
cells
nodes and the
will
spleen.
enter the thymus,
And
or those cells that
then we
enter the
have
thymus will
where
become
we
T
get
cells.
the
known
clonal
as the name T
selection
cells.
occurring.
And those that enter the bone marrow will become B cells. Both of these cells, both the B and the T, will develop antigen receptors on their cell surface. And this is really an amazing thing because neither have yet to be exposed to the antigen, but they will develop those antigen receptors. And this process itself is called clonal diversity. The lymphocytes, both the B and the T lymphocytes, will then leave the primary lymphoid organs as competent but yet naive cells. And what that means is they have never been exposed to the antigen, but they will be able to identify the antigen. And for this process, which occurs in the clonal diversity to happen, there are some secondary entities that have to play a part in this. The cells that are necessary to help with this would be those lymph organs, such as the spleen and the lymph nodes. And what will happen is once these cells enter the blood and the lymph vessels, and they migrate to the lymph organs, which will be the spleen and the lymph nodes. What I would like to do is use this illustration, and as you look at this illustration, I want to explain to you the two phases of adaptive immunity. So the first phase, which is clonal diversity, it begins when we are fetus in our mother's womb. And it is in the thymus and the bone marrow, which are the primary lymphoid organs. So what happens is cells enter the thymus and they become T lymphocytes, also known as T cells, or they become B lymphocytes, known as B cells. The T lymphocytes come from the thymus, the B lymphocytes come from the bone marrow, hence the names. they both will develop antigen receptors on the surface of the cell. Like, this is an amazing process in our body because it develops antigens to something it has never been exposed to. And this process alone is known as generation of clonal diversity. The lymphocytes, both the B and the T lymphocytes, which are developed, will then leave the primary lymphoid organ as competent but yet naive cells. And what this means is that these cells have never been exposed to the antigen, but however, they are able to identify those antigens. And for this process to actually occur, the help of the secondary organs are necessary, the lymphoid organs. And these organs that are needed to help with this process are the phagocytic cells and the primary dendritic cells. And what will happen once they help facilitate this process, the cells will then enter the blood and the lymphatic vessels, and they're going to migrate to these secondary lymph organs. And these are the spleen and the lymph nodes. So as you see in this picture, you can see how it goes from the primary lymph organs to the secondary. And then what will happen is these cells will then reside either as B cells or T cells in these organs. And what we believe right now is that 60 to 70% of the circulating lymphocytes are immunocompetent T cells, while 10 to 20% of those are the immunocompetent B cells. And so that is phase one, the clonal diversity phase. Then we have phase two, the second phase, which is clonal selection. So what happens with clonal selection? In clonal selection, it is initiated once we have a foreign antigen. So that's what initiates the second phase. What will happen is the antigen reacts with the T and the B clones and the cells. And these are the things, the clones and the cells have these antigens on their surface. Remember, we said that they have built their surface receptors, right? Which is against those specific antigens. And thus it begins to initiate further differentiation and proliferation into new mature effector cells. This requires the cooperation of the secondary lymph organs and the antigens to help in that process. And then what will happen is those antigens will then be used, and those antigens used, why do I have? I would like to now utilize this slide to talk you through the process that occurs, or the two phases of adaptive immunity. We're going to start talking about the clonal diversity stage. With the clonal diversity stage, it begins when we are still in our mother's wombs, when we are fetuses. And what occurs is in the thymus and the bone marrow, which are the primary lymphoid organs, we will have the development of T lymphocytes, which comes from the thymus, and B lymphocytes, which comes from the bone marrow. And they will both develop antigen receptors on the surface of the cell. Now, this is an amazing thing because one of the things that is amazing about this is that these cells have never been exposed to the antigen. However, they can build up receptors to antigens. And what we call this process is generation of clonal diversity. And so then the lymph nodes, both the B and the T lymph nodes, will then leave the primary lymphoid organs, which is our thymus and our bone marrow, and they will be considered competent but naive. And what that means is these antigens, or these lymphocyte cells that have never been exposed to the antigen, so they're considered naive. However, they're competent because they can recognize the antigen. In order for this process to fully be completed, although it occurs in the thymus and the bone marrow, there is the need for secondary organs to use the phagocytic cells and the primary dendritic cells to process these antigens. And then once that is done, the cells can then enter the blood and the lymphatic vessels, and they can migrate now to the secondary lymph organs, which would be our lymph node and our spleen. And when they're in the lymph node and the spleen, the cells will stay either where the B cells sit, which would be in the spleen, or where the T cells sit in those organs. It is believed that about 60 to 70% of the circulating lymphocytes are immunocompetent T cells, while 10 to 20% are our B cells. So that is your phase one. And then in your second phase, the clonal selection, what happens is we get an initiation or exposure to a foreign antigen. Once that exposure happens, the antigen will then react with the T or the B cell, or the T or B cell clones that are present. And once they have that interaction, there will be specific surface receptors specific to that antigen. And thus it can start to initiate a differentiation and a proliferation of those cells into mature effector cells. And then once that process happens, it will require the cooperation of the secondary lymphoid organs and the antigens to help process that as well. And again, we think about the phagocytic and dendritic cells that will help with that process. And then the antigens used in this process will be referred to as our antigen presenting cells, or what we refer to as APCS. And this leads us to cellular interaction between the APCS subsets of the B and T cells. And then we get intracellular adhesion through the antigen receptors and specific intracellular adhesion molecules. And they then produce the response of multiple cytokines, and eventually we get further differentiation of the immunocompetent B and D cells, which now become highly specialized effector cells. And then the B cells develop into plasma cells that produce antibodies, while the T cells develop into the T cytotoxic cells, and it helps to regulate the clonal selection, which we think of those cells as the TH cells, or they can suppress and limit the immune response. So we also have T regulatory cells as well. So the T cells could be either the clonal selection cells, the TH cells, or we can get the immune response, the ones that suppress the immune response, which are our T regulatory cells if necessary. However, both the B and the T cells, although they separate out and do their separate jobs, they both will then form and differentiate and have some memory cells. And that's the difference. Remember about adaptive immunity, where they have memory, whereas when we talked about inflammation, it has no memory. Now, if that was very confusing for you, there are going to be further slides talking about each one of these cells separately, but please refer back to your textbook that will help give you clarity. Now let's talk a little bit about the two arms of our immune system, the humoral and the cellular. And what this is these are the arms of our immune system in which antibodies and T cells both fight to protect our bodies against infection. And they do that by having the antibodies that circulate in the blood. So when we are exposed to any type of microbial toxins or pathogens, that there will be an immune response that would be able to fight off that infection within the blood. So the next few slides, we're going to talk about what components make up this part of your immune system and how does it work to fight off infection and how they work together to do this. As you look at this slide, what I want you to see is that when we talk about these two arms of our immune system, humoral and cell mediated, which cells help to protect us and how they do that. So if you look at humoral, our primary would be our B cells. And then if you look at cell mediated, it would be T cells. But what's important to note is that although they work together, they do have some differing responsibilities. So make sure that you are aware of how each one of these entities become effective in fighting off immunity in our bodies.
Now, to conclude our discussion on general characteristics of adaptive immunity, I'd like to talk briefly about active and passive immunity. Now, when we think about active immunity, this is the immunity that is produced by individuals after natural exposure to an antigen or after they've been immunized. But contrary to that, we have passive immunity. And passive immunity does not involve the host immune system at all. However, it is a response that occurs and that is performed when antibodies or T lymphocytes are transferred from one donor to a recipient. And an example of this would be when a mother passes antibodies to her unborn child, to her fetus. Now that we've concluded our discussion on some of the general characteristics of adaptive immunity, we're going to do a quick knowledge check. So there'll be a few questions that you can read, and then you'll have the answers that will precede the question, and you can kind of see and check your knowledge base. And if you're having difficulty with these questions, I would tell you to make sure that you go back and review the information in your textbook. And then once you complete the few knowledge check questions that we have here, we are then going to move now to the section I like to title Recognition and Response of the Adaptive Immune System. Now let's talk about some of the aspects of the recognition and response. What we know when it comes down to the immune system, an effective immune system is an immune system that can specifically recognize an antigen by antibody or receptors on the surface of the B and T cells. And it's followed by a set of complex intercellular communications among a variety of the APCs and the lymphocytes. Antigens are the molecules in which the immune response is directed. There are two terms that we refer to when we think about antigens: antigen and then immunogen. They are often used simultaneously, although they are not exactly the same. They do have some clinical differences that are important to note. So when we're talking about an antigen, we are talking about a molecule that can react with binding sites on antibodies or antigen receptors on the B and the T cells. But when we talk about immunogens, they will induce an immune response, resulting in the production of antibodies or functional T cells. So not every substance that is an antigen is immunogenic. So that's important to note. Also, when you think about the binding that occurs, and it's referred to as what we call antigenic determinant or the epitope. And then there's a portion of the antibody or the lymphocyte receptor, which is referred to as the antigen binding site or the paratope. So you have the binding site, the paratope, and then you have the antigen determinant site, which is the epitope. So these are just some terms that it's important to kind of remember when you're thinking about the immune system regarding antigens. When attempting to consider if an antigen is immunogenic, there are criteria that should be considered. The four criteria are if the antigen is foreign to the host, if it is the appropriate size, does it have the adequate chemical complexity And is it in sufficient quantity? When thinking about tolerance and antigens, historically we used to think that tolerance meant that there was a state of non-responsiveness to which the immune system would passively allow self-antigens to persist. We have now evolved in our understanding of tolerance to include a self-tolerance and peripheral tolerance. The previous slides, we discussed antigens and immunogens that play a role in the recognition of the adaptive immune system. Here on this slide, we identify the 3 molecules that help to recognize antigens.
So now that you've reviewed what an antigen is, let's talk a little bit about antibodies. There are a diversity of antibodies. There are 5 major antibodies that we normally think about. Antibodies also can be called immunoglobulins. And we have the classes of IgG, IgA, IgM, IgE, and IgD. In the next few slides, we will talk about each one of these individually. We're going to first look at the IgG antibody. That's going to be the first one that we talk about here. This is one of the most abundant antibodies that we have, and it is the antibody that is transmitted from mother to fetus through the placenta. When you think about IgA antibodies, think about the antibodies that is predominantly in your blood. And this antibody normally can be found in body secretions as well. Now, when you think about IgM antibody, think about the antibody that usually is the first one to be produced after being exposed to an antigen. It is also the antibody that is synthesized during fetal life. Now, IgD is one of the most limited of the immunoglobulins that we talk about, and in the lowest concentration in the blood of all of the immunoglobulins. Now, IgE, just as IgD is normally can be in low concentrations, but IgE is most known because it usually is the most common immunoglobulin seen in allergic reactions. Now, here's a chart that I have put together, and what it does is it shows you side by side the different immunoglobulins and what their functions are.
Although there are a few more slides that we are going to use and to discuss more about recognition and response of adaptive immune system, I would like to take a minute to do just a knowledge check. We've kind of gone through quite a bit of material here, so we're going to do a quick knowledge check. In order to understand how antibodies work, one must have a good understanding of the actual molecular structure of an antibody. The structures, which consist of 3 parts: the antigen binding fragment, the crystalline fragment, and the polypeptide chains. We discussed in the previous slides how antibodies' molecular structure is important in understanding an immune process. It is also important to understand how the antigen actually binds to the antibody to work as a complex for our immune system. So in the next few slides, what we're going to talk about is that actual complex, the B cell receptor complex and the T cell receptor complex. And if you remember when we discussed earlier in the lecture about the complexes and how there is actually the formation of the T cell and the B cell, This is just giving you more detail about that process. We discussed in the previous slides about B cells and T cells, how they are structured, how they build complexes, but now we also need to discuss and think about the molecules that help to present these structures or to the antigen. Let's talk a little bit about what happens to cells when it comes down to transplantation. Now, during transplantation or after transplantation, what we know is that a recipient's immune system can respond or have a response against the major histocompatibility complexes. Thus, we want individuals as much as possible to be compatible with the donor HLA tissue. The more compatible they are, the more likelihood that that transplant will be successful. Now, we talked earlier about how B cells are matured in the bone marrow. The next few slides are going to give you the specific information of how that maturation actually occurs. In the previous slides, we talk about the maturation of the B cells. Now I want you to read the next few slides that explain how T cells mature. We have already discussed 3 areas regarding adaptive immunity. The first was general characteristics of immunity. The second was recognition and response of adaptive immunity. The third was the first phase of adaptive immunity, which is known as generation of clonal diversity. We will now transition to discuss the second phase of adaptive immunity, which is clonal selection. When you think about clonal selection, there are several aspects that you need to think about regarding clonal selection. One of those would be secondary lymph organs, organs such as the spleen, lymph nodes, the adenoids, and tonsils. And we talked about that a little bit earlier in this lecture. It is important to understand some of the functions of T cells. Functions such as how they activate our macrophages, as well as how they help with regulation of lymphocytes. I would like to conclude this lecture on the adaptive immunity by talking about some of the effector mechanisms of adaptive immunity. One of those would be the ability of the adaptive system to protect against infection. And one of those ways it does it is through neutralizing these antigens. And this system can also do it either directly or indirectly. Also, the adaptive immune system has the ability to produce or utilize the IgE immunoglobulin in a manner that is not common to what we think about when we think about IgE. When we think of IgE, we think of allergic reactions. However, what the adaptive immune system is able to do with IgE is that it can recognize that there are certain parasites that invade our mucosal tissue. The process that occurs with the B cells can switch the IgE into secreting plasma cells, which is under the direction of the Th2 cells, and thus primarily produce the IL-4 cells. And what all of this does is it allows the IgE to now fight off the parasites. So there is a process that occurs with IgE, which is initiated by the adaptive system. So these are just a few things of that we see related to the adaptive immune system, but I wanted to just highlight some of that for you. So this will conclude my lecture regarding the adaptive immune system. There is much more to be learned about the adaptive immune system, and there is much more that you can even study in your textbook if you would like. But I am going to stop here because you have gotten a lot of content. My recommendation in summing up this lecture is go back to, I believe it's slide 2, which is the outline that shows you the big concepts that you need to grasp and understand related to the adaptive immune system.